This topic is about morphologic patterns of acute inflammation, which is one of your final exam questions. And an easier way to know this topic, to put it is basically classification of inflammation by exudate, which is the dominant phase of acute inflammation. This will make you understand how to go about it in the question. So let's go. So just a quick review of the classification of the phases of inflammation. The inflammation usually starts with ulcerative phase, moves towards the exudative phase, and then ends with the proliferative phase. Ulcerative is usually any kind of damage caused to the tissue, from any minor to any severe changes, for example, steatosis or something till necrosis, for example. Exudative, which is the main topic for today, exudative inflammation, which is the most prominent phase in acute inflammation, and also describes the morphology of the acute inflammation, is the second phase of most inflammation processes. And the third phase, proliferative phase, it is the most important phase in chronic inflammation. So it is not the topic for today. But it's good to know that, just a quick review, that it consists mainly of the lymphocytes, fibroblasts, and involves processes of repair and regeneration. But what's important to remember is, along with the exudative inflammation, you also have the 5 typical features of inflammation, which are mentioned here. So it's always necessary to remember these features and never forget them. Okay, right. These are just a few pictures just to show how an alteration might look like. Basically, any damage to the cell, to the tissue. But yeah, so exudation. Well, basically, it's exudation. It is the release of liquids and proteins from blood vessels damaged by the inflammation, and later on as well, the blood cells as well. So, an exudate is mostly made up of proteins, water, and some blood cells. But there are different types of exudates, so depending on the type of exudate, we know what it consists of, or what is the composition of an exudate. Right. And why does inflammation result in an exudation process? That is because of vasodilation, which sort of pushes out the plasma and the proteins and the blood cells from the vessel into extracellular spaces. Okay, so the 6 main types of exudates are serous, purulent, non-purulent, fibrinous, and gangrenous. And it's very important to remember all of them with examples for each. And we'll go through each one by one now. So what's important to know is knowing all the names, the description, what they consist of, how they look, as in the color, basically. I mean, they're liquid, but the color is more important here. And examples for each, right? So let's start with the serous exudate. It is mainly watery kind of exudate, consists of transparent watery liquid, mostly made up of plasma, and it's very poor in cells and protein. It's the simplest type of exudate, you could say, and it's mostly, these are the most common examples which are to be remembered. That's why I put them in red. So it's good to remember the examples according to the surface you're talking about. For example, you could divide each exudate from the surface it comes out. For example, deep and superficial. And then if we're talking about superficial surfaces, we can talk about mucosa and skin mainly, as well as serosa. And then deep, we talk about interstitium. So the most common examples for serous inflammation in superficial tissues are acute rhinitis, herpes vesicles, and from the serosal surfaces, pericarditis, pleuritis, etc. And from the interstitium, we can talk about urticaria. So giving an example to your examiner gives him or her the idea that you know the topic quite well and you understand what you're talking about, and not just mugging up stuff and saying it from the book without an understanding. So it's a good idea to go about it. The gross morphology of serous inflammation would be a watery, transparent fluid, for example, in the form of vesicles. In the case of skin, herpes vesicles, or just transparent fluid like acute in the case of acute rhinitis. Also, what's good to know is, in some kind of inflammation, it is in most kind of inflammation, it is serous exudate is the initial step before any other exudate appears. So it can be either standalone, like in the cases of these examples, or it can be for a slight period be the initial exudate. For example, in the case of some bacterial infections, which we'll see in the upcoming slides. Right. So here's an example of a situation where serous exudate is only present in the initial stage, and then later the prevalent exudate or inflammation takes over. So this is an example of erysipelas, which is an infection caused by mainly Streptococcus bacteria and causes purulent inflammation. Right. So next is purulent or suppurative inflammation or exudate. You can call it either, because the reason why we call it both is because inflammation, the type of inflammation is basically defined by the type of exudate in the case of acute inflammation. Hence, you can call it either purulent inflammation or purulent exudate. Right. So what exactly is purulent inflammation? It is an exudate consisting of pus, and the color of the pus can be from yellowish to greenish, and it is mostly made up of dead neutrophils and necrotic cell debris, and mostly it is from due to bacterial infections. Right. We give a basic description. We give a basic macroscopic morphology. Now we move on to the examples, which will complete your answer in an exam. So for the examples of purulent inflammation, we can divide them into superficial and interstitial, or superficial and deep surfaces. So for the superficial ones, we have bacterial peritonitis, bronchopneumonia, and pustules, which are made in the case of impetigo caused by staph or strep bacteria. And here's a picture of impetigo, how it looks like. These are basically pustules. So like yellow like bumps on your skin, you could say in simple English. For the example of deep purulent inflammation, we have abscesses, phlegmon, and empyema mainly. There's a basic description of all of them, which will help you remember and remind yourself what exactly it is. Right, so abscess is a localized collection of pus that forms a cavity. And it's a good way to describe it in the form of liver abscess caused due to parasites, for example, or lung abscess caused due to aspiration, or when we talk about phlegmon, I saw a picture in the previous slide. It is so for phlegmon, a very classic example is erysipelas due to Streptococcus bacteria. And what exactly is phlegmon is? It's a diffuse spread of purulent inflammation in loose fibrous connective tissue without sharp demarcation, like mentioned over here. This is in contrast to abscess, which is a cavity and with pus localized to the cavity and in one space. So it's interesting how these two things are different. And last is empyema, which is presence of pus in preformed cavities. And what are the preformed cavities? Well, your pleural cavity, your gallbladder, your pericardium, all these are preformed cavities, which weren't there, which have always been there since birth. Right. So this is about purulent inflammation. If you remember the points on these slides, for the exam, basically. Next is non-purulent, or also known as lymphoplasma cellular inflammation. I will quickly go through this one because it is mainly found in chronic inflammation rather than acute, but we need to mention it since it's part of the question. So the predominating cells are the B cells and the plasma cells here. And the very classic examples for this kind of inflammation is from the mucosa, you have chronic bronchitis or the chronic sinusitis. From the serosal surfaces, we can describe viral meningitis. From the classic example, contact allergy, which is a chronic situation. From the interstitium, we couldn't talk about mumps. Okay. Next is gangrenous or putrefactive inflammation, very important, also very dangerous. So this is the kind of exudate with wet gangrene. And what exactly is wet gangrene? Well, it is a necrosis modified by putrefactive bacteria in severe alteration. So the putrefactive bacteria causes the necrosis to be modified and infected, and it makes it look like sort of like green, grayish color. A picture in the next slide. And the putrefaction consists of cell debris and putrefactive bacteria and necrotic cells, basically. Morphologically, it exhibits massive necrosis without demarcation, and it can be a life-threatening situation in some cases. For example, in the case of gangrenous appendicitis, it is a surgical emergency. The other example is lung gangrene caused due to tuberculosis or cancer. Example to remember here is gangrenous appendicitis. And this is the picture of the gangrenous appendicitis. Look how the gangrene looks greenish, grayish in color. And this is the acute appendicitis, which further, due to the presence of putrefactive bacteria in the feces, becomes gangrenous appendicitis. So this is how gangrenous appendicitis develops. Okay. Last, we have the fibrinous inflammation. It is one of the most important kind of inflammation which you need to remember very well, and also it's very confusing. So it's good to several times. All right. So what exactly is fibrinous exudate? Well, fibrinous exudate is when the permeability of the vessel is so much that it allows a fluid containing fibrinogen to be to come out of the vessel basically, and become part of extracellular matrix, and it becomes fibrine over there. It often happens in the body cavities, in the pericardium or pleura, but also in meninges, for example. How it looks like morphologically and microscopically. It's whitish grayish deposit and sort of hard, and it's followed by organization, which is a dangerous phase because if it becomes organized, it becomes fibrotic and causes adhesions, and in some cases may also obstruct the organ and compromise its function. And why do we have such fibrosis and adhesions? Because wherever you have fibrine coming out, it will initiate organization. So fibrine and organization, they always go together, and it's not a very good situation. It's a pretty bad situation because the organ's function is being compromised. And we'll see some examples, and I'll explain how how the function becomes compromised. What's important to note here is which cells are involved in this process of fibrosis and adhesions. The fibroblasts and macrophages are mainly involved in this process. Okay, right. So the examples for the serosal surfaces, we have the fibrinous pericarditis. Now imagine if you have fibrin in your pericardium, and if it becomes organized, you end up with some adhesions. And how will your heart function anymore properly if you have adhesions in your pericardium? Um, how will it contract? How will it pump anymore? The second example for fibrinous inflammation is skin and mucosa, um, which is which consists of pharyngitis of diphtheria, for example, and pseudomembranous colitis caused due to Clostridium difficile, which is a very important and common example. And this is how it looks like, pseudomembranous colitis, and it is a bad situation if it gets organized. But what's if we want to have a favorable situation, then we should have a lytic phase, basically the breakdown of the inflammatory exudate. If that happens, then we're happy. But if it organizes and fibrosis happens, then it's not a nice situation. The last example for fibrinous inflammation is from the interstitial surfaces, basically exudation on the inner surface of the pulmonary parenchyma in the case of lobar pneumonia, especially in the gray hepatization stage, which is the third stage of lobar pneumonia. It's good to revise the stages of lobar pneumonia to understand this example. So you have deposition of fibrin in the alveolar walls, and if you have fibrin in the alveolar walls, and if it gets organized, you lose your alveoli basically. It doesn't function anymore. So yeah, and some more examples are rheumatic fever, SLE, scleroderma. Okay, what's important is I mentioned about pseudomembranous colitis. What exactly is a pseudomembrane? So it's very important to remember that these four contents make up the pseudomembrane. And when you describe a pseudomembrane, you need to talk about these four, otherwise your answer is not complete. So remember, a pseudomembrane is made up of inflammatory cells, necrotic cell debris, fibrin exudate, and microbes, and most commonly present in diseases like pseudomembranous colitis and pharyngitis of diphtheria. And it is not a true membrane, hence the name pseudomembrane. Right. So this is a quick review. I wouldn't say quick. I mean, it's a detailed picture of all the with all the examples of exudative inflammation, and you do not have to remember all of them. But for each kind of exudate, it's good to know one example from for each kind of surface. For example, you have serous inflammation. You remember some example, one or two examples from superficial inflammation, and you remember one or two examples from deep. Similarly, for lymphoplasmacellular exudate, remember a few examples from superficial and some from deep. Most examples are from the superficial, basically, so it's good to remember more from the superficial inflammation than the deep ones. And what makes it easier is that you remember just the most common examples rather than trying to sit and memorize this whole chart, because trust me, it's not going to work. I tried it, it didn't work. You can't remember the whole chart when you have so much to study. So just remember the most common examples for each classification, and you should be good to go. And without examples, your answers in pathology will never ever be complete. So please remember the examples when you're studying for the exam.